Today we'll be going over the predicted paper for Biology 2024, paper 1, since it's coming up in a few days. It's this Friday. So we'll start simple. So question 1, the diagram below shows a single cell algae which lives in fresh water. Here is the diagram shown. So A, which part of the cell labeled above traps light for photosynthesis? If we look up, this is the chloroplast. This is because it has something called a chlorophyll, and this chlorophyll is a pigment and it traps light for photosynthesis. So which part is made out of cellulose? The only thing made out of cellulose is the cell wall, and it's this part. This is only unique to plant cells. B, in the fresh in water environment, water enters the algal cell. What is the name of the process by which the water moves into the cell? So as soon as we see water, we already know that it is osmosis, since that is what is the transport movement of water. In any type of way, it is always osmosis. This is for AQA hires, though some things will be relevant to trilogy. Okay, so I give the reasons why the algal cell does not burst. Why does it not burst? So since we know that it has a cell wall, this cell wall is the reason why it does not burst, because this cell wall is really strong. So it's not able to burst easily because it kind of pushes this out, so it's not able to move the water. So this is the reason why, it's because it has a cell wall. So in the marking, we could just write cell wall, but it's better for you to understand what you said. So cell wall allows it not to burst. C, the algal can photosynthesize. Complete the word equation for photosynthesis. This is something that you just have to learn. So it is water and plus CO2. This is carbon dioxide. And this slight energy goes into glucose and oxygen. For respiration, it's actually the opposite way. So photosynthesis and respiration, it's both the different ways. So photosynthesis, the arrow would be going this way, while photosynthesis, the arrow is this way. So once you learn one equation, it's kind of the same as both equations. So I, the flagellum helps the cell to move through the water. Scientists think that flagellum and the light sensitive spot work together to increase photosynthesis. It's just why this might happen. So if we read the question carefully, so it helps water move through the water. We know that that's osmosis. Scientists think that flagellum and light sensitive spots work together in photosynthesis. So this is because the light sensitive spot, this detects light. So this is the first point of what photosynthesis happens, because it's not able to happen without light, since that starts the reaction. And then this tells the flagellum to move towards the light. This is the flagellum over here. It's kind of a tail, and the function of this is so that it can swim and it can move to different ways. So this tells it, the sun goes here, it tells it, this means that the whole, it's like the brain of the thing, and then it's able to go and move. So therefore it moves, and then more light equals more photosynthesis, since the light will go in, and it will start this reaction over here that we just put. So D, multicellular organisms often have complex structures such as lungs for gas exchange. Explain why single-celled organisms like algae does not need complex structure for gas exchange. Why they don't need is because they are small. They're really small things, so that they don't need such complicated structures like how we have, because we are much bigger than them. So we need to be able, so that we can reach all the gas, the oxygen, the carbon dioxide, so that it can reach all the different parts of our body. We have these complicated structures because they're just a small thing. Their large surface area to volume ratio is very big. Unlike ours, it's small. So it's a shorter distance. So if we get all the good things that come out from here and it goes into there, it's so much easier going to get all the space in such shorter time. like in compared to like a bigger one, this is going to take more time to fill with a gas that we need. That's why it's so much better. And with these type of questions with gas exchange, make sure that you always put large surface area to volume ratio. That's the key word that they're always looking for, even though you explain it right. But with GCSE, the main point is really to make sure you say what the mark scheme requires. So then you just kind of link it back to the question and what it's asking. So you say, though, that means the cell membrane is good enough. Okay, question 2. All living cells respire. A. Respiration transfers energy from glucose to muscle concentration. Describe how glucose from small intestine is moved to a muscle. So we know it starts in the small intestine. How does it move to a muscle? So anything moved in the body, this is always by blood. So we know that's the first one. So we know that glucose, how is it from small intestine? How does it go into blood? This is always going to be by diffusion. You have 3 modes of transport. This is diffusion, active transport, and osmosis. Osmosis is with water. So we know that's not the answer. We know that it's going to be diffusion because it goes from high concentration to low concentration, which is what's happening here. And then the blood will go to the muscle cell. So it's absorbed by diffusion into the bloodstream. And then the blood delivers the glucose to the muscles in the capillaries. Blood transfers it everywhere. So B. The diagram below shows an experiment to investigate anaerobic respiration in yeast cells. What is the purpose of the liquid paraffin in tube A? So if you see here, we want to see anaerobic respiration. So you can see here, this is the cell. What the liquid paraffin actually does is it stops air from getting in so that there's no oxygen, and with oxygen, it will make it aerobic respiration. So that we can make it anaerobic, we need to stop any oxygen or air coming into it so that we can make sure that they respire anaerobically. So this is what the liquid paraffin does. It stops any of these gas bubble and air to go in. Stop air going in. C. The indicator solution in tube B shows change in the concentration of carbon dioxide. The indicator is blue when concentration of CO2 is very low, and green when the concentration of CO2 is low. Yellow when the concentration of CO2 is high. What color would you expect the indicator to be in tube B during the maximum rate of anaerobic respiration? Maximum rate of anaerobic respiration, so if you know what yeast anaerobic respiration, when it respires, what it does, the products of it is CO2. So we know that if more it anaerobically respires, we know CO2 is what's going to come out from it. So lots of CO2 will be here, and that's what actually makes the gas bubbles. So we know it's going to be very high, therefore yellow. And we kind of know, even if you don't know and you just want to guess, when you see very low and low, it's very hard to differentiate between it because it didn't really give us a range or number. Therefore, it's very subjective, so it's more likely that yellow will be correct. D. Suggest how the experiment could have been changed to give a reproducible way to measure the rate of reaction. So what this can be is that it can include any apparatus. So what I put is that it collects CO2 with a gas measuring cylinder, and volume is collected in a certain time using a timer. Rate of reaction is always amount over time. And because it's amount over time, we need to find a way to get amount and over time. So time is always going to be using a timer, using a watch, and this is just over a certain amount of time so that we have, so let's say over 30 seconds, over a minute. And the amount, how we are going to find the amount is collected using a gas measuring cylinder. This can also be done using a gas syringe. So over here, you can either put a syringe, so it's like that, and it kind of collects all the gas collected, or you can do a syringe where it kind of looks like that, and you kind of get it all. Please let me know if any of you guys have any questions in the chat, and please share and follow for more everyday questions. So compare anaerobic reaction in yeast cells with anaerobic respiration in muscle cells. So what this is just a compare question. We need to make sure that we always put a similarity and a difference. How we can put this is that we are going to put what the equation of in yeast cell anaerobic respiration, what's the equation in muscle. So this is the equation in yeast cell. It's glucose goes into ethanol and CO2, and energy is the byproduct. That's why we do it. And in muscle cell, it's glucose and it creates lactic acid. So what we can say is the similarity is that both of them release small amounts of energy. That's the similarity. However, the difference is that we can see that yeast produces CO2. This does not have CO2. And yeast also produces ethanol, while glucose produces lactic acid. You can also say that yeast cell has 2 products are formed, while glucose only has 1. Question 3. The photograph shows the same cell of a common pod plant. Photograph A shows the cell in a hypertonic solution, while photograph B shows the same cell in a hypertonic solution. So the first question is, what is a hypertonic solution? So I would recommend from the beginning, once you're reading the question, is that you make sure that you understand the question, because understanding the question really help you answer the questions and know what they're looking for in terms of the mark scheme answers. So what is a hypertonic solution? You can see from the hyper I always like to think as more. If you're very hyper, it's more. So it's a solution more concentrated than the cell. You just need to remember that we're talking about the solution. So that means hypo, it's less, is when the solution is less concentrated. So this is the 2 marks come from its more concentration, and this is the second mark comes from then the cell. B. What word is used to describe a plant placed in a hypotonic solution? In a hypotonic solution, all the water will go out. No, in a hypotonic solution, all the water will go in, so this is called turgid. Well, in a hypertonic solution, all the water will go out, so it is flaccid. Or a different word for this is plasmocide. So you can choose whichever you can remember more. I like to remember flaccid. So this is because as it goes through osmosis, and water goes from where it's from a high concentration of water to a low concentration of water, or you can say it goes from a low concentration of solutes to a high concentration of solutes, so that they can balance the ratio between water and solutes and whatever thing is dissolved in it. So, okay, so C. Explain what has happened to the plant cell in photograph B. So if you go back to photograph B, we can see that over here, we can see what the cells look like. We can see what has happened to it. So you can see that the water has left from cell A. They have left from cell A, and so we always need to describe, if we're talking about water or anything, we need to describe what actually has happened, and how it happened. So since we know it's water, we know that it is osmosis. So water left the cells by osmosis from a more dilute to a more concentrated solution via a partially permeable membrane. So just describing what osmosis does, even if you don't know what actually happened and you don't know the question, but you see that they're talking about osmosis, you can just describe and just define what osmosis is, and you'd already get 3 marks. So this means that the cell membrane shrank away from the cell wall, as you can see here. So you can see here, it shrank, and therefore it's going, so that you can kind of see that how it's going, while here you can see that there's a lot of water. That's why it's so much more visible. So here it is. You can stop or screenshot it so that you can have it. D. Animal cells will also change when placed in different solutions. Some red blood cells are put into hypertonic solution. Describe what would happen to these red blood cells and explain why this is different to what happened in plant in photograph A. So what is a hypotonic solution? That is when it's low. When there's low, so that means there's a lot of water, so that means that water will enter the cell by osmosis, because that means that the cell has less water than the outside. remember when we talk about this, we're talking about the solutes. So Walter insert, this means that it burst. This is because red blood cells kind of look like this. They do not have a cell wall. Instead, they have a cell membrane. So animal cells have no cell wall, and cell wall is what prevents it from bursting. These are the marks that you get so that you can get 4. So just to paraphrase it, is that the water will go in, and that means it will burst. And the reason why it bursts is because there is no cell wall, and it's the cell wall that actually does prevent it from bursting. So question 4. The diagram shows a strawberry plant, and the parent plant grows on the side shoots, and new plants grow on these side shoots. So the new plants will all have the same inherited characteristics as the original plant. Complete the sentence to explain why and use the words from different blocks. So let's go with A. The new plants is produced by what type? So we can see that it has all the same inherited characteristics. So that means that it is by asexual reproduction. Because it's asexual reproduction, this is because we saw the word same, and because it's same, we know that it's genetically identical. So genetically identical is made from asexual reproduction. Sexually makes non-genetically identical daughter cells. So it is not the same. So in these type of reproduction, body cells divide by, this is by mitosis. This is because, again, mitosis is what makes it so that it's all genetically the same. It is meiosis that makes it non-genetically. So the new plant has the same genes as the parent. So this all go hand in hand. So I recommend that you learn it completely. So, and then if we just go over these ones, embryos are all, they are from meiosis, they're from sexual reproduction, and from fertilization and differentiation. This is just a process in which embryos go through, as well as gametes in them. So it is mitosis, genes, and asexual. This is all because when you see that they are from the same genes and the same genetically identical type, they're all genetically identical, they all have the same inherited characteristics. This can only be done with these 3 different types. Question 5. A woman gives birth to triplets, and 2 of the triplets are boy and a 3rd is a girl. So the triplets developed from 2 eggs released from the ovaries at the same time. The diagram shows triplets A, B, and C developed. So stage 1, 2, 3, this is how diagrams show. Which stages on the diagram shows the gametes? If we go here, it is 2 and 3. Because the gametes are the egg and the sperm. So you can see that these are the egg and this is the sperm. So they go and then they form the gametes. So it is 2 and 3. B. Embryo B is a male. Which of the following explains why embryo B is a male? So if you look here, embryo B is a male. Why? This is because cell P has a X chromosome and cell R has a Y chromosome. So it's XY, and as we know, XY is a male while XX is a female. So this one. C. The children that develop from embryos A and C will not be identical. Explain why. And you may use the words from the box in your answer. So they will not be identical. This is because they are formed from different eggs, and meaning that they have different genes. So if you can see here, A and C are they're from different ones. So over here, these are formed from C, P, and R, while this is formed from Q and S. So these have different genes altogether, while A and B will be identical. Single cell from an embryo at stage 7 can be separated and grown in a special solution. what term describes what are grown in this way? And these are in stem cells. What happens when the cell are placed in a special solution? This is what happens is that the cell divide, and then they divide, and then they differentiate. But divide comes first. So give one use of cells grown in this way. So one way that I just think that you should memorize is medical research. And this is because they're able to grow so many different types of cells. And because they grow all these different types of cells, it turns into they're able to use it for medical research instead of just using a person. So IV. Some people might object to using cells from embryos in this way. Give one reason why. Answer is always the same when they talk about object. It is because there is ethical obligations. This can be religious. This can be moral. Some other reason is that it can also potentially harm the embryo. And it also could be that like you're playing God, which is why there's like religious obligation and stuff. So my way is that they will ask for one reason is just to put ethical objections. Question 6. Microorganism can cause infection. So the human body has many ways of defending itself against microorganisms. So describe 2 ways in which a body prevents the injury of microorganisms. So question 6, microorganism causes infection. The human body has many different ways of defending itself against the microorganisms. Give 2 ways in which the body prevents the injury of microorganisms. So this can be that the skin forms a barrier, and so it doesn't allow different types of viruses to go in. Also, the acid in the stomach, HCl, kills all the pathogens, bacteria in the food. It can also be the hairs in the nose traps and particles which may contain pathogen that stops it from entering through your nose. And it could also be that trachea, bronchi has mucus and this traps pathogen, and also the cilia in the throat, so it prevents anything from being swallowed. B. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before this new drugs can be used and to treat people. This is 6 mark, and this is the same thing over and over again. It's the way that every type does. So what happens is before you always start with preclinical trials on the new drug and cells. So this can be on tissues, or it can also be on live animals. So the whole point of this is to test toxicity and the dosage, its efficiency. So what type of dose do you need before it becomes, so if it doesn't work at all, it's too low. If it works too much and causes a lot of side effect, it's too high. And then once you kind of know and start to see that it's pretty efficient, then you move on to clinical trials. And this is where you start testing on healthy volunteers, and you can test it on Ebola patients. So what you do is that you could do a double-blind trial where the healthy volunteers or the Ebola patients don't know if they're getting a placebo or if they're getting things, so that it doesn't allow them to see to start mimicking side effects even though it's not real. So you know how efficient the drug actually is. And you can also do a random allocation of Ebola patients to groups, so no one actually knows. And then what you do is that you do the trials and you start at very low doses. So this allows you to monitor the safety and the side effects. And then you only do trials to find the optimum dosage and testing for efficiency. And once you do this, you peer review data with different scientists to make sure, and it helps prevent any false claims. And then from there, it can actually be used to treat people once it's actually effective and has gone through all these procedures. Question 7. Why blood cells protect the body against pathogens such as viruses and bacteria? Pathogen makes us feel sick. Give one reason why. So one reason why is that it produces toxins and poison into the body. So like as if you're getting food poisoning, that makes you very ill. And it also, that causes damage to the cells. So it does that by killing white bacteria, like blood cells and different cells that you have. So that's why sometimes your skin might be the one that's affected, is damaging skin cells. 2I, white blood cells produce antibodies, and this is one way white blood cells protect us against pathogens. Give 2 other ways in which white blood cells protect us against pathogens. This is what they do. They produce antitoxins. So pathogens produce toxins. It's kind of like a counteract is antitoxin. So that kind of neutralizes whatever acid, whatever thing they're putting onto the bloodstream. What they also do is that they engulf and ingest pathogens. So let's say this is the pathogen. What it does is that it kind of goes on top of it and it kind of surrounds it and circles it. And that way it kind of eats it. So it's kind of like Stuvia. So, and this is called phagocytosis. Though it's a really fancy word. You don't really need to know it, but it does help you prove that you know what you're talking about to the examiners. So B, vaccination can protect us from the disease pathogen causes. I, one type of viruses causes measles. A doctor vaccinates a child against measles. What does the doctor inject into the child to make the child immune to measles? So this is basically what they're asking, even though they gave us so many different type of information. What the question is asking us is it's asking, what do you put in a vaccine so that it can work? And what the answer always is dead virus. So you get 1 mark for dead. And this can also be inactive or weakened, a weakened type. And the second mark is for virus, or it can also be pathogen. And that gives that two word gives you 2 marks. So when you try and answer a question, see what exactly the question really wants from you, and that's where you start getting the marks, so that you know what the mark scheme looks like, because that is the trick with GCSEs. A few weeks after vaccination, the child becomes infected with measles virus from another person. The graph shows the number of measles antibodies in a child's blood before the vaccination and after the infection. More measles antibodies are produced after the infection than after the vaccination. Describe other differences in the antibody production compared to with after. So it says describe. So describe does not mean explain, which is what a common mistake people do. They try and explain why this is happening. That just wastes your time and it wastes mark. We are just describing, so we have a graph. We use data from the graph and we just say what it is in words. So what it says is that antibodies are produced after are more, and then what it is that if you see after the infection happens, you can see that the number of antibodies that are in the body is much more than when you just first got it. So it's much more steeper and it's a faster rise in that number. So that means it also is way longer lasting. If you see over here, it stays such a low steep in comparison to here. So here it goes down pretty fast in comparison. The time is kind of the same, but here it's so much longer. So what we can say is that after the infection, the rise begins sooner. So that means there's less lag time. This over here, where it's actually the exact same antibodies, this is called a lag time. So it takes, that's why maybe you might have got sick, but you don't actually feel the symptoms a day after. So the last time meaning also faster rise, this therefore, as well as longer lasting. 3I, vaccination against measles viruses will not protect the child against rubella virus. Why? This is always the same thing, whether they talk about enzymes, whether they talk about immunity and viruses, whether they talk about pathogens. The answer is that antibiotics are always specific. It's like how enzymes are specific. It's like how proteins are specific. Antibodies are proteins. C. What is the advantage of vaccinating a large portion of the population against measles? So because it says large portion, this is talking about the reduce of spread of infection. This is what we like to call herd immunity. So they're not talking about what the benefits are for one person. They are saying a large portion of the population. So it reduces spread of infection. Make sure you look closely on what the question is actually asking you before you try and answer. Question 8. Enzymes are used in body cells. What is an enzyme? So draw a ring around the correct answer. So here, an enzyme is a catalyst. What is a catalyst? It is a biological catalyst. So what is a catalyst? It speeds up the reaction of anything without actually being used up. And that is what enzyme does. It speeds up the reaction. It allows protein to be made. And it's like the lock and key theory as well. So 2I, all enzymes are made up of the same type of substance. What is the substance? This is protein. And that's why when I said in the previous question, proteins are specific, that means enzyme are, antibodies are, they're all the same. But enzymes are not the same as antibodies, don't make that mistake. So where in the enzymes amylase produced in the human body? So it is produced in the salivary glands. It's one of the first ones that come out, and that breaks down starch. So B, enzymes are sometimes used in the industry. Draw one line from each enzyme to the correct industrial use of that enzyme. So carbohydrates, it changes starch into sugar. So you can just see from carbohydrates, starch is a carbohydrate. That's one way that you can find it. Protease, so you can see that it's protein, so it says pre-digest protein in some baby's food. And with iso, this is glucose. So glucose syrup into fructose syrup. You can kind of do it, so how I did it was that I did the ones I know, and then you kind of just see between these two. Isomerase, it's thing. As soon as you see A's, you know that it's an enzyme. Question 9. The leaves of the most plants have stomata. Name the cell in which controls the size of the stomata. These are the guard cells. So the stomata is kind of like that, and these are the guard cells. This allows it to either relax. If it relaxes, it goes in, or it can contract. So if it like squeezes, so that means it goes out and it opens. So that is what controls it. Give one function of the stomata. The stomata has a lot of different functions, but the one that I like to remember is that CO2 to enter. However, different ones that you can have is that it allows the control loss or evaporation of water, like controlling the transpiration rate. It can also allow oxygen to leave. Basically, it's a function for plants for their gas exchange, how we have our lungs. So B, the image below shows a part of the surface of the leaf. The length and width of the piece are both 0.1. Calculate the number of stomata per leaf. So if you see here, 0.01, so we can count how many. There is 2 stomatas. These are the stomatas. So if you see over here, these are the guard cells that I was talking about. And the one that is in the middle is a stomata. So what we have to do is that we have to times it to see how many, how much is this area, so that we can see. So 0.0.1, that is 0.01. And if you see here, there is 2. So you do this by 2, so it is 200 per millimeters, because you do this per meter. You want to see how per millimeter, so times this by 2, 0.02, and you divide it by that. So 2 eyes. A different plant species has 400 stomata per millimeters of leaf surface. Having a large number of stomata per millimeters of leaf surface can be a disadvantage to plant. Give one reason why. So what we usually think it's an advantage. It's an advantage because we get more light in, and this is very good for photosynthesis and for CO2. However, what it also does is that there's more water loss. And this increase of water loss means that it's more likely to wilt. It's more likely to like sag because of it. That's why we want to keep the water inside so that they're able to use it instead of just completely losing it. So C, a student investigated the loss of water from plant leaves. The students did the following. Step 1, took 10 leaves from a plant. Step 2, weighed all 10 leaves. Step 3, hung the leaves in the classroom for 4 days. Step 4, weighed all 10 leaves again. Step 5, calculated the mass of water lost by the leaves. mass water loss. Step 6, repeated steps 1 to 5 with a grease spreader on the upper surface of the leaves. Step 7, repeated steps 1 to 5 with a grease spread on both of the upper and lower surfaces of the leaf. So as you can see here, I was kind of just highlighting all the important information that I think I need to remember. It kind of just makes me make a bit more sense of what's happening so that I can answer the questions faster and more efficiently. So the table below shows the students' results. So here it shows the treatment, so the different types, and also the mass water loss in the leaves. Okay. So question 1, what mass of water loss was lost in 4 days through the upper surface of the leaf? So we can see here, we can see that we can just minus, so it is 0.12. So very little water was lost when the lower surface of the leaves were covered in grease. So explain why. So why is that the lower surface has the most stomata? So when you look into a leaf, it is this point of the leaf that has the most stomatas, not the top, it's the bottom. So stomatas are now covered, meaning that the water cannot escape. And because the water cannot escape, meaning that there is less water loss, this is what the stomata is the function for. So question 10. The plant exchange substance with the environment. Plant roots absorb water mainly by osmosis, while plant root absorbs ions mainly by active transport. Explain why roots need to use 2 different methods to absorb water and ions. So this is because in the beginning, when it first happens, is that solution in the soil is more dilute than in the root cell. So this means that it has more water. So this means that water moves from dilute to the more concentrated region, which is osmosis. This is the definition of osmosis, goes from dilute to more concentrated. And then, so why do we use active transport? This osmosis does not use energy. So concentration of the ions in the soil is less. This is because the plant is always going to want more ions so that it can help it. So it always takes it up from the soil. So more energy is needed so that they can move the ions. Since this is the opposite, it has a low concentration in the roots, but high concentration. So it goes from low to high, meaning it's active transport, meaning it needs energy. B, what is meant by the transpiration stream? So this is just something that you need to memorize. It's the same 3 marks all the time. It is the movement of water from roots to leaves via the xylem. So it's a movement. So what we get water from, here. And this moves up the xylem, since xylem is what carries water, the phloem is what carries sugar, so that it can go up to the plant. So here we go, we describe the direction. And the last mark comes from that water evaporates from the stomata, where the stomata are down there. So it evaporates from it, and therefore it's making that stream, so it's getting that pull so that it can go up, kind of like how when you suck a straw. So C, students investigated the loss of water vapor from leaves. So the student cuts up some leaf, measured the mass every 30 minutes, so from 180. So the rate of mass of water, 30 minutes was 7, and calculate the rate. So what is rate? Rate is amount over time, 0.67. So 2i, the rate of mass lost between 0 and 30 minutes was very different from the mass of that loss between 30 and 180. So just an explanation for the difference between 2 rates. So during the first 30 minutes, it was warmer, so there was more evaporation. And because it's warmer, more evaporations happen so that it can cool itself down, kind of like how it's sweating. So question 11, an athlete carried out 6 months training program. So graph 1 shows the effect of the same amount of exercise on his heart rate before and after the training program. So you use graph 1 to find the heart of the trained athlete 5 minutes after the start of the exercise. So this is 5 minutes. So you want to find, after the start of the exercise. So if we go here, this is the exercise, so we need 5 minutes after. 5 minutes will be over here, so we go from 10 and we go up, and we can see over here it's 120 since they want after, so this is 120. So if you can see here, lots of students mistake by putting up over 5. This is why it's very important to know what the graph is talking about and to make sure that you know what the data is so that you can make sure that you get these simple marks very easily. The stroke volume of the heart is the volume of blood pumped out of the left side of the heart in one heartbeat, and the graph 2 shows the relationships between stroke volume and heart rate before and after the athlete did the training program. This is the key. So cardiac output is defined by heart rate times stroke volume. So clearly show your answers out. So calculate the cardiac output of a train 5 minutes after the start of the exercise and use your answer from part A and thing. So we know it's 120 heart rate. So what we want to see is that we want to do 120. So we got to go from here. 120. So we want to see after the start. So we go over here. We go there. So it's around 98. So it's heart rate times stroke volume. Stroke volume is 98, what we figured out. So the cardiac output is 11760. So graph 1 shows that from the same amount of exercise, the heart of the trained athlete was beating more slowly than it did before the training program. You just explain why. So this is because the trained athlete has a higher stroke volume and more blood per beat. C. An increased cardiac output will provide more oxygen and more glucose to the working muscles. So explain why this helps the athlete during the exercise. So this helps it. This because when you see that something to do with exercise, a really good rule of thumb is that you follow these answers no matter what the question is. So an increased cardiac output will provide more oxygen, more glucose. Oxygen and glucose, these are the reactants of aerobic respiration. This means there is an increased aerobic respiration, meaning increased energy supplies. So this means that it delays anaerobic respiration since they're making enough energy supply, meaning less lactic acid formed. And we know lactic acid causes cramps. And once you get a cramp, it's really hard for you to actually continue with what you're doing. So that you're having them and you're delaying them, this means that you can work harder and work faster and longer. Make sure when you see these type of questions where it's explained, you always link back to the question about what they're specifically asking you. That usually is the last mark. Question 12. A. Complete the word equation for photosynthesis. So carbon dioxide, water, glucose, and oxygen. So this is just something that you need to memorize. Respiration is the other way, where these are the reactants and these are the products. B. Draw a ring around the correct answer to complete each sentence. So the energy needed from photosynthesis comes from light. While respiration, this is for humans. Osmosis is from water transport, how water transports. Energy is absorbed by a green pigment called chlorophyll. So chlorophyll is inside chloroplast, but the pigment itself is called chlorophyll. So we need to make sure that you know the difference between them. Chloride is just a chemical, it's not that. If temperature is decreased, the rate of photosynthesis will decrease. This is because temperature, it's directly proportional. So temperature increase, photosynthesis will also increase, which is the same as vice versa. And this is just because temperature is very closely correlated to light. And that's the end of the paper. If you want to try this paper, it's on Stuvia in our account, Study Scholars. If you looked on our pinned video, then you can find it. So thank you, everyone. If you have any questions, please let me know, and we'll be happy to help.